Welcome guys, this is Joyce Angel from First MSc Applied Microbiology, Sacred Heart College, Tirupattur. In this video, we are going to study about biochemical tests. These biochemical tests are classified into enzyme-based biochemical test, carbohydrate-based test, protein-based test, and in-week test. In these biochemical tests, now we are going to study about enzyme-based biochemical test, establishing enzymatic capabilities. Enzyme-based tests are designed to indicate the presence of a single enzyme as well as a complete metabolic pathway. In this, we are going to identify whether the specific enzyme present in or not. Some of the enzyme-based tests are catalase test, oxidase test, coagulase test, phenylalanine deaminase test, and urease test. Catalase test. The aim of this catalase test is to differentiate the bacteria that produce an enzyme catalase such as Staphylococcus species from non-catalase producing bacteria such as Streptococcus species. In this, the Staphylococcus species has the ability to produce the enzyme catalase, whereas Streptococcus species do not produce catalase enzyme. It is based on the principle that the enzyme catalase catalyzes the release of water and oxygen from hydrogen peroxide. If you see in this reaction, this catalase enzyme reacts with hydrogen peroxide to form water and oxygen, which results in bubble formation. Materials required: test bacteria, sterile glass slide or test tube, 3% hydrogen peroxide solution and glass rod. Procedure: in tube catalase test, 1 to 2 ml of 3% hydrogen peroxide solution is poured into a test tube. Using a sterile glass rod, several colonies were taken from the fresh culture. The culture should be 18 to 24 hours old and it is immersed in the 3% hydrogen peroxide solution, and then it is observed for immediate bubble formation. In slide catalase test, use a sterile wooden stick or glass rod to transfer a small amount of colony in the surface of clean glass slide, and then place a drop of 3% hydrogen peroxide in the glass slide. After that, observe for the evolution of bubbles. Interpretation and results: if the given organism is catalase positive, it will produce the bubbles as you see in this picture. If the given organism is catalase negative, it will not produce any bubbles. Example for catalase positive organism is Staphylococcus species, and example for catalase negative organism is Streptococcus species. Oxidase test. The aim of this oxidase test is to detect the enzyme cytochrome oxidase in gram-negative bacteria which produce this enzyme such as Pseudomonas, Neisseria, Campylobacter and Pasteurella species. Principle: the cytochrome oxidase test uses certain reagent dyes such as phenylene diamine dihydrochloride, which is impregnated in the oxidase disc and that acts as a substitute for oxygen as artificial electron acceptors. It is colorless in the reduced state. Bacteria which contain cytochrome C as part of their respiratory chain are oxidase positive and turn the reagent into blue or purple in color. Organisms which are lacking cytochrome C do not oxidize the reagent, hence leaving it colorless within the limits of the test, and they are oxidase negative. If you see in this reaction, cytochrome oxidase enzyme reacts with tetramethyl paraphenylene diamine dihydrochloride and turns it into purple or dark blue in color. Materials required: test sample. Here 24 hours culture is recommended for better results. Oxidase disc, glass slide, sterile wooden stick or glass rod. Procedure: pick the bacterial colony by using sterile wooden stick or glass rod and dip on the surface of oxidase disc. Do not pick the inoculum with a metal loop because the oxidase reagent may react with the metal and it may cause false positive results. After that, observe inoculated area of oxidase disc for a color change to deep blue or purple within 10 to 30 seconds. Interpretation and results: if the given organism is oxidase positive, purple color will appear within 30 seconds as you see in this picture. If it is negative, no color change is seen beyond 30 seconds. Example for oxidase positive organism is Pseudomonas species. An example for oxidase negative organism is Escherichia coli. Coagulase test. The aim of this coagulase test is to determine the ability of the organism to produce coagulase which clots plasma. Some of the Staphylococcus species produces coagulase enzyme. By using this test, we can distinguish the pathogenic coagulase positive Staphylococcus species from the non-pathogenic coagulase negative Staphylococcus species. It is based on the principle that the specific Staphylococcus species produces coagulase enzyme and that converts soluble fibrinogen into insoluble fibrin. There are two forms of coagulase are found. One is bound coagulase, otherwise known as clumping factor. It is detected in the coagulase slide test and it can directly convert soluble fibrinogen into insoluble fibrin and causes the staphylococci to clump together. Free coagulase. It is detected in the coagulase tube test. It reacts with the globulin plasma factor, that is CRF, coagulase reacting factor, to form a thrombin-like factor. Staphylothrombin catalyzes the conversion of fibrinogen into insoluble fibrin. Materials required: glass slide, distilled water, rabbit plasma, test organism and pipette. Procedure: in slide coagulase test, place a drop of saline on each end of a slide or on two separate slides. With a sterile loop or straight wire or wooden stick, emulsify a portion of the isolated colony in each drops to make two thick suspensions, and then add a drop of human or rabbit plasma to one of the suspensions and mix gently. After that, look for clumping within 10 seconds. In tube coagulase test, dilute the plasma in physiological saline, that is, mix 0.2 ml of plasma with 1.8 ml of saline. Then take 3 small test tubes and label as test positive control and negative control. Test is 18 to 24-hour broth culture. Positive control is Staph aureus broth culture and negative control is sterile broth. Pipette 0.5 ml of the diluted plasma into each tube and then add 5 drops of the test organisms to the tube labeled test and 5 drops of Staphylococcus aureus culture to the tube labeled positive control and 5 drops of sterile broth to the tube labeled negative control. After mixing, incubate the 3 tubes at 35 to 37 degrees Celsius. After that, examine for clotting after 1 hour. If no clotting has occurred, examine at 30 minutes intervals for up to 6 hours. Interpretation and results. In slide coagulase test, white fibrin clots are observed. As you see in this picture, it shows the positive result. Smooth suspension indicates negative result. In tube coagulase test, formation of fibrin clots indicates positive result and no clot formation indicates negative result. Coagulase positive organisms are Staph aureus and Staph intermedius, and coagulase negative organisms are Staphylococcus epidermidis and Staphylococcus saprophyticus. Next, phenylalanine deaminase test. The aim of this test is to differentiate the Proteus, Morganella and Providentia species based on their ability to produce phenylpyruvic or pyruvic acid by oxidative deamination from other members of the Enterobacteriaceae family. It is based on the principle that the deamination of amino acid results in a colored compound with the addition of 10% ferric chloride. If you see in this reaction, here the enzyme phenylalanine deaminase reacts with phenylalanine and forms phenylpyruvic acid. The presence of phenylpyruvic acid is detected by adding 10% ferric chloride and it forms green color. Microorganisms that produce phenylalanine deaminase remove the amine group from phenylalanine. This reaction results in production of ammonia and phenylpyruvic acid. The phenylpyruvic acid is detected by adding few drops of 10% ferric chloride, which acts as a chelating agent. Here, chelating agent means compound that reacts with metal ions. A green colored complex is formed between these two compounds, indicating a positive test. Materials required: test bacteria, phenylalanine agaslan, 10% ferric chloride, incubator and inoculation loop. Procedure: prepare a phenylalanine deaminase agar medium. Inoculate the test bacteria by streaking the phenylalanine agar slant. Incubate the inoculated phenylalanine agar slant at 37 degrees Celsius for 24 hours. Following incubation, apply 4 to 5 drops of 10% ferric chloride solution directly to the slant. Then gently agitate the tube and observe for the development of green color within 1 to 5 minutes. Interpretation: formation of intense green color within 5 minutes after applying 10% ferric chloride were observed in the positive result, as you see in this picture. If it is negative, the slant remains its original color. Example for positive organism is Proteus mirabilis. An example for negative organism is E. coli. Finally, urease test. It is used to determine the ability of an organism to produce the enzyme urease, which degrades urea, and it also used to identify the rapid urease producers such as Proteus and Morganella species, and weak urease producers such as Klebsiella and Enterobacter species. It is based on the principle that the urease enzyme reacts with urea in the presence of water to form carbon dioxide and ammonia. This ammonia combines with carbon dioxide to form an alkaline compound, ammonium carbonate, which results in increased pH and turns the indicator phenol red from its original orange-yellow color to bright pink color. Procedure: streak the surface of Christensen urea agar slant with well isolated bacterial colony and incubate the inoculated agar slant at 37 degrees Celsius for 24 hours and then examine for the development of pink color. In case of unknown result, incubate the tube for 7 days to check for slow urease production. Observation and results: if the color changes from light orange to pink rapidly within 1 to 2 hours, it indicates the positive reaction. Example for positive organism is Proteus species. For less active species like Klebsiella, it may require 3 or more days to show positive result. If it is negative reaction, the agar slant retains its original color. Example for negative organism is E. coli and Enterobacter species. Thank you for listening.